All right, let's dive right in. We're going to tackle a question today that honestly sounds like it's straight out of a sci-fi movie. What if we could actually program life? I'm talking about a technology where biology and computer science are crashing together, and it's making all of this a reality right now.
No, seriously, just pause and think about that for a second. Imagine you could sit down at your computer, write a few lines of code, hit a compile button, and design a living, breathing cell to do exactly what you told it to. It sounds wild, right? Well, that is the huge idea we're digging into.
For the longest time, genetic engineering has been, well, it's been more of an art than a science. It was this super specialized craft. It meant years and years of trial and error in a lab. You had to have this deep, almost gut level knowledge. But this new way we're talking about, it completely flips that on its head. It turns it into an automated, engineered process, a lot more like writing software. And just to give you an idea of how big a deal this is, to put it in perspective, it wasn't that long ago that a researcher said it took a postdoc, a highly trained scientist, 3 full years just to design, test, and build one of these biological circuits. 3 years for one! I mean, that's just an incredibly slow and painful process.
And that right there, that was the big problem. The potential was absolutely huge, but getting there, man, the process itself was this massive, massive bottleneck.
So you had to have this deep, intuitive feel for how all these different genetic parts were going to play together inside the incredibly messy, chaotic world of a living cell. It was way more guesswork than it was predictable engineering.
But then, something changed. Researchers over at MIT had this huge breakthrough. They created what you can basically think of as a compiler for life. And listen to how the lead researcher, Christopher Voigt, described it. He said, and I quote, "It is literally a programming language for bacteria. This isn't just a metaphor. You sit down, you use a text-based language, you're basically coding, just like a programmer would for a computer."
Okay, so how on earth does this actually work? Well, the process is surprisingly straightforward. Step 1, you write your program. You use a text language. It's actually based on Verilog, which is usually for designing computer chips. Step 2, the software takes your text and compiles it. It translates your code into a real functional sequence of DNA. Step 3, you take that DNA and pop it into a living cell, something like E. coli. And finally, step 4, the cell just runs the program. The new genetic instructions kick in and do their thing. The lab bench has effectively become a keyboard. It's wild.
Okay, but the magic, right? How can a cell understand computer logic? I mean, it's not made of silicon. Well, the secret is all about building logic gates, but with biology. The key concept here is something called a synthetic biological circuit. So think of an electronic circuit in your phone, right? But instead of wires and silicon chips, this circuit is built out of genetic parts, things like genes and promoters. And you assemble them inside of cells so they can perform logical functions, like sensing something in their environment and then responding to it. And this is where it gets really cool. You can actually design these biological parts to act just like the logic gates in a computer. So, for example, you can build a sensor that detects, say, glucose, or you could build an AND gate, where the cell will only do something if and only if it senses both signal A and signal B, or an OR gate, where it'll act if it senses signal A or signal B. And this is literally programming decision-making into the DNA of a living thing. Okay, so that's the theory. But you're probably wondering, does this stuff actually work in the real world? Well, the initial results were pretty mind-blowing. So get this, the research team designed 60 different biological programs using this new language, and out of those 60, 45 of them worked perfectly on the very first try. That's a 75% success rate right out of the gate. Think about that. A process that used to take years was now happening almost overnight with this incredible level of success. So what does this mean for us? What can we actually do with this kind of power? Well, the possibilities are just staggering. I mean, imagine programming bacteria to hunt down cancer tumors and deliver medicine directly to them. Or yeast that can monitor its own fermentation process and shut it down if things go wrong. Or even programming the microbes in your gut to help you digest lactose better. We're talking about a complete game changer for medicine, for manufacturing, for everything. Look, this isn't just some new lab tool. What we're witnessing here is the dawn of a completely new field. We're really entering the age of biocomputation. And if you look at this timeline, you can see just how fast things are moving. The basic science really got going in the 2000s.
Then, boom, 2016, you have this breakthrough programming language. Now, in the 2020s, we're already seeing things like AI-driven drug discovery taking off. And when you look ahead to 2030 and beyond, it seems pretty clear that programmable biology is going to be central to, well, to everything.
And the impact is going to be felt across the board. We're talking about personalized medicine designed just for you, sustainable manufacturing that doesn't rely on fossil fuels, even using DNA for data storage, which is insanely dense. Not to mention smarter farming and way faster, more accurate medical diagnostics.
All of this is coming together to create what people are now calling the bioeconomy. And it's exactly what it sounds like, a future where engineering life is a main engine of economic growth, and maybe our best shot at solving some of the world's biggest problems, like climate change or future pandemics. And that really brings us to the big final question. For basically the first time ever, biology is turning into a platform that we can program. So the question isn't, can we do it anymore? The question is, what should we do with it? As we get better and better at writing the code of life, what are we going to tell it to do next?